So we've just talked about how is the cell membrane formed, what is its structure, and now we're going to look into the functioning of the cell membrane. So how does that form? How do those phospholipids and those proteins and those molecules affect how it functions? When we talk about the functioning of the cell membrane, one thing that we want to keep in mind is this second law of thermodynamics. Okay? Now the first law of thermodynamics says that energy or molecules, matter, can't be created or destroyed. And the second law, it says that all those molecules, the liquids and gases, are going to be constantly in motion. And they're actually going to move towards disorder. Another word for this is actually entropy. Entropy, okay? Now, when you're thinking of this, you can think of gas molecules, okay? If you have your one fellow student that's sitting on the desk next to you, that's suddenly in the front of the room and has sprayed all over with perfume or with Axe, a can of Axe, sprayed all over, and all of a sudden you're in the back of the room, you see this happening, you see those molecules of Axe, but eventually, about a minute or two later, if you're really far across the room, you might start to smell it, okay? And that's because those molecules are constantly in motion. They're not going to want to stay in that one area of high concentration, but instead they're going to want to move towards the area, right over here, where there's a low concentration of those molecules, or no concentration of those molecules. And so that movement from high to low concentration is known as diffusion. Okay? And diffusion is going to occur because molecules are going to want to spread out. All molecules, liquids and gases, are going to want to spread out until the concentration gradient, I'm going to do a bracket for concentration. The concentration gradient is equal. And when this happens, it's going to be known as equilibrium. So concentration gradient is just the difference of where the molecules are higher to where they are lower. What's that difference in between? Another thing you want to keep in mind is that molecules are going to want to keep moving and keep spreading out until they hit some sort of barrier. And that barrier could be something like our cell membrane. When we're thinking about the cell membrane, because of those properties of the phospholipids and because of some of the proteins that are there, there are some molecules that are going to be able to diffuse easily. And those molecules are going to be small, they're going to be nonpolar, or they're going to be uncharged, no charge, no ions, okay? Molecules that can do this easily would be like oxygen gas, O2, and carbon dioxide gas, CO2, okay? Those are going to move through really easily. Now, some other things that can move through the cell membrane easily would be lipid-based molecules, like some hormones, can also move through very easily because of their chemical properties. Now, when we talk about transport, there's two main types that we'll want to focus on, and that's going to be active and passive transport. Okay? So some rules here about active and passive transport. On the left-hand side, we're going to have our passive transport. And passive transport is going to require no energy. You're going to be moving from a high concentration, H with the bracket, to a low concentration. And the other thing is that you're actually going to be moving with the concentration gradient. Now, concentration gradient, I want you to think of that hill with a wagon at the top. When that wagon's at the top, you're moving high to low, that's going to be really easy, smooth sailing. You're not going to need energy to get that wagon down the hill. Now, some proteins are also involved in passive transport. So diffusion is a form of passive transport, but there are also some proteins that assist in this. Then we have some that are called channel proteins. Channel proteins are actually going to be very specific. All proteins are, and they're going to be shapes. They're going to be specific with their shape and charge. An example here would be an aquaporin. Aqua, okay, is actually going to allow water to pass through. Water doesn't just move through those phospholipids. Water actually has to go through a channel protein. And the channel protein is very specific in allowing it to move through and not needing energy for that to happen. Then you have your carrier proteins, and your carrier proteins are also going to be very specific. Your carrier proteins are going to actually have a conformational change. Now, what does that mean? Conformational change, thinking back to our proteins and our enzymes, that's a shape change. And so here you have a molecule or a solid moving into the cell membrane. The protein itself shifts or conforms, produces a different shape, and releases that molecule inside. And then it'll go back to its original form, or at least when it needs more of those molecules to come in. Now, sometimes this can require energy, but sometimes it doesn't. The last type of passive transport is actually two things. It's going to be exocytosis, the movement of molecules out of the cell, and endocytosis on the other side. In exocytosis, basically you have a little packaged membrane called a vesicle, has molecules inside of it. It could be waste, and it's actually going to move out, get really close to the cell membrane, and merge with that cell membrane, CM. Okay? And it's merging. When it merges with it, it's actually going to merge and then push those substances, those molecules, out. And that's what you see happening in the picture. Now, the opposite can happen. You can have endocytosis, where you have molecules outside of the cell that need to get in. They can get really close to the cell membrane, but not be able to move through the cell membrane through proteins or through the lipid bilayer. And so instead, the cell membrane will actually pinch around it, and the opposite would happen. Carry it into the cell, and eventually that vesicle that's been made will actually release those substances needed. So in endocytosis, your cell membrane is actually going to surround those molecules, and they're going to go inside of the cell. Okay, pretty easy. So again, keep in mind that all three of these are going from high to low concentration. You're not going to require energy. On the opposite hand, we have active transport, and active transport is going to move from low concentration to high concentration. Okay? And this is going to need energy. Now, in most cases, active transport is going to require some sort of protein or pump, okay? And it's actually going to be collecting molecules. So if you think about low to high, you're at the bottom of the hill in that wagon, you're moving upward, that's going to require a lot of energy to get that wagon up the hill, especially if someone's sitting in it. Now, when I talk about collecting things, a lot of times this means that you need more of something. So let's say somebody threw a bunch of money in the air, and you're trying to collect all of it. That's going to take a lot of energy for you to run around and pick up all that money. That's kind of what's happening here. to use energy. Notice that ATP is used, moving from high to low, and there's actually something called a coupled transport that can occur. Coupled transport, there's two types, symport and antiport. In symport, that means that the molecules are actually going to be moving in the same direction. Notice they're all moving one way into the cell. Antiport means that they're going to be moving in the opposite direction. Some are moving in and some are moving out. Okay? But what we really need to be able to do in this unit is be able to predict how are the molecules or water going to be moving. Sometimes it's going to be solute stuff, and sometimes it's going to be water. But the movement of water is known as osmosis. And osmosis is going to move from high to low concentration. So here you have an example of this U-shaped tube. On one end you have a hypertonic solution, a solution, solutes plus water. Hyper, usually if you're really, really hyper, you think you're someone being high on sugar, okay? So that means you're going to have a high concentration of solutes. When you have a high concentration of solutes, that means you're going to have a low concentration of what I'm going to call free water. Remember that when you have a molecule inside of water, inside of a solution, the water molecules, because of their polarity, are going to try to surround that cell or that molecule. And when it surrounds it, it gets kind of stuck there. It wants to stick there and be there because of those chemical properties. On the other hand, you can have a hypotonic solution where you have a low concentration of solute and a high concentration of that free water, H2O. Okay? And so when you're looking here, these little molecules, the solutes, remember this is just stuff inside of the solution. It can be like salts, it can be like sugar. And if you have a high concentration of solute on this side and you have less free water, we're going to be wanting to move towards equilibrium. We want to move from high to low concentration. So in these types of tubes, only the water can move. And the water, there's going to be a net movement of water from high concentration to low. Eventually, the water is going to reach a point of equilibrium. Now, equilibrium, when you think about this, this is going to mean that the concentration on both sides of water and solute is going to be the same. Now, when I say that, it doesn't mean it's going to be the same in number, but that it's going to be in the same ratio of water molecules to solute molecules. The last thing you want to do is to be able to predict the water's effect on cells. If you're looking at this first cell, if this first cell is in a hypotonic solution where there's a low amount of water, that means that it's going to have a high amount of solute. Okay? And so you want to think about what's going to happen. Well, in this image, they're showing you that the water's rushing in. And what's the effect of that? Well, the cell, the animal cell, can actually lyse or break. With the plant cell, it's going to be a little different because they have that cell wall for extra support. So try to make those predictions on what will happen with the cell and its membrane. And then tomorrow, we're actually going to figure out a little bit of math on how to determine and make those predictions mathematically.